Sure. I'm Evan Tjeerdema, and I'm a PhD candidate at Scripps Institution of Oceanography at UC San Diego.
What inspires you to do the research that you do?
Maybe I should back up a second and first preface like the kind of work that I do is I work at the interface of environmental toxicology, cell biology, and marine biology. And I think where that comes from is first from early childhood, like most things, I suppose. I have kind of this longstanding interest in the oceans and in the natural world. And as I got older, I became more aware of some of the environmental problems that are facing marine ecosystems and marine organisms. And through college, it was really OChem where things changed for me. I took OChem, and most people don't like OChem, but I had a really good time in OChem. And what that kind of way of thinking with organic chemistry is that everything is very mechanistic, and there's a cause and effect, and you can pinpoint specific chemicals or specific reactions that have certain outcomes. And I really found that interesting and fascinating that there are these mechanisms underlying issues with toxicology and issues with pollution that we see. It's not just a black box of some poisons that we don't know what they are, and then something happens that we don't understand, and then there is death or some kind of toxicity or birth defects, things like that. But you can actually like open that black box up and dissect it and figure out kind of like how and why toxicity happens in, say, certain animals or following certain exposures or in certain tissue types. And so that the way that I kind of get from there to like my graduate research is understanding not just like the mechanism of the chemicals, but also the mechanism of the biology. Marine organisms and cells, they aren't just like kind of passive entities in the environment. Like they have ways of protecting themselves and mitigating toxicity from compounds. And so I was really fascinated by the idea of like being able to dissect the biology a bit and figure out, well, what can the organism do to protect itself, and how does that determine the toxicity that we see, and what are those interactions like, and what happens, maybe most interestingly, is like what happens when those defense mechanisms that an organism has becomes compromised. It starts really from these like early in life kind of interests that were then fostered, but then as you get older and you're exposed to kind of more fields and more things, and you have the opportunity to think about these problems in different ways, a little bit more like mechanistic and nuanced, and I find that really interesting. So it's like personally and professionally very satisfying.
Maybe you could say a little bit about the major highlights of what you're going to be presenting on.
Sure. Yeah, I'm happy to talk about that. So the project that I'm presenting on really, I think, is quite a step forward in understanding this phenomena that we see in environmental toxicology. And the phenomena is called chemosensitization. And just very briefly, what that is that there are chemicals in the environment, like these synthetic persistent organic pollutants, things like DDTs and PCBs, that are long-lived and they're not very easily broken down. And what they actually do is that inside of cells after an exposure, rather than being very effectively cleared by some of like the xenobiotic metabolizing systems that your cells have, what they can actually do is they persist in spite of that. And one of the reasons is that they're able to actually bind and inhibit the activity of a particular class of xenobiotic metabolizing proteins called drug transporters. The drug transporters are proteins that sit in the membrane, and what they do is they actively efflux xenobiotics out of cells to prevent them from building up and prevent them from causing toxicity. What these persistent compounds do is they actually get into cells and they'll bind and inhibit, and they're not transported, but they just lock in and they sit there, and they basically block the transporters from effluxing other small molecules that are toxic. And so that causes chemosensitization because it actually increases the potency of a second compound that the transporter would otherwise be moving because it's blocked. But there's a gap in knowledge that's been there for 30 years since this was first proposed, which is that we don't know what those secondary molecules are. Like we've had a lot of insight, particularly in the last 5 years, but for a couple of decades, that synthetic compounds in the environment can interact with drug transporters and perturb them and can inhibit them. But we haven't known what the additional toxic substrate molecules are in the environment that are becoming more toxic because they cannot be cleared as effectively. Like what drives toxicity once a transporter is inhibited. And so the work that I'll be presenting is actually like our efforts to figure out what those are. And we have pretty good evidence now that some of these compounds are actually they're actually molecules that are produced naturally in the oceans by marine microbes as secondary metabolites and intermediates in those biosynthetic pathways. And so the work that I'll be presenting on is essentially we developed a pipeline to screen through these compounds in sea urchin bioassays for toxicity with and without drug transporter activity, so we can see if the animal gets more sensitive or if there's no difference when it doesn't have the drug transporter, kind of like a positive control. And so what we were able to do is we were able actually to screen through a library of purified marine microbial product and detect them for toxicity, but most importantly to detect them as whether or not they were more potent to animals that don't have a functional drug transport.
How do you create like a living organism that doesn't have these transporters in them? Like, wouldn't it? I just feel like the organism would like die or something because it's like it sounds so critical.
Yeah, that's a good question. It is definitely critical, but it's not essential for survival. For a couple reasons that drug transporters are redundant. So on the cell surface, it's not just one protein that's doing all of the work at protecting the cell from toxic small molecules. There's a whole assortment. There's a whole assortment of different drug transporters in there. I mean, the family of just the family consists of about 390 different genes. So there's a lot of proteins there. That being said, the drug transporter that we targeted is the gene ABCB1 or P glycoprotein. In the sea urchin embryo in the early life stages, that one gene accounts for about 30% of all of the efflux activity. So even with the other transporters that are present at the same time, ABCB1 seems to be doing the lion's share of substrate efflux. And so that's why, like, if you target the specific gene, like, you do see a difference because it accounts for such a large portion of the transport activity that we observe.
Right.
But to your first question about how do you make an animal that lacks this protein. So to make a knockout line of a gene of ABCB1, we used CRISPR-Cas9 mutagenesis. And what's really nice about sea urchins is that they're actually really amenable to genomic engineering and gene editing, because people have long since figured out how to do transfection, how to get these reagents into the early life stages, like into the egg. And like anyone who works with sea urchin knows that they produce hundreds of thousands of eggs pretty much on demand, and they fertilize externally. And you can work with them under a microscope really easily without too much specialized equipment. And one of the things that you can do with them under a microscope is this technique called microinjection, where you take a tiny glass needle and essentially inside this needle is a mix of reagents that you want to use to perform gene editing. So for CRISPR-based gene editing, you would have, like, inside this mix, what we use is an mRNA that encodes for the protein of Cas9, and then some guide RNAs that target the protein to the specific DNA sequence in the animal that we want to target. And so just under a microscope, you actually go through and you take this needle and you individually poke these zygotes, and you deposit a tiny volume of these reagents actually inside the egg, and the egg seals back up, and then it progresses through development as normal. But then a subset of what we've injected will have the mutation, and so then you just grow the animals up and you screen them out for the mutation that you want, and then you pick the founders and then you propagate those, and so then all the offspring, all the hundreds of thousands of sea urchin offspring, will have that same mutation, that same CRISPR-Cas9 deletion in the gene. And so doing gene editing in sea urchins is actually really powerful because you get thousands of offspring to work with that all have the same gene edit that you're interested in. So it's a bit more powerful in a sense than other model organisms like mice or even a zebrafish, where you can be limited. You can certainly be limited by the number of organisms of individuals that you have to work with, right? And what we have now is that then you have parents that are full knockouts for this gene, and so then you can just spawn them and perform your bioassay on the embryonic stages, and then compare with a wild type, and you can see the effect of a chemical on the animal that does and does not have the drug transporter, and really robustly evaluate the influence of the drug transporter on protecting the embryo from that particular compound.
How did you first get connected to SoCal SETAC?
So when I was an undergrad, SETAC North America was the first conference that I presented at. I think I was a junior in undergrad, and I went to UC Davis, and SETAC happened to be coming to Sacramento that year, and so just up the road, it made perfect sense. I heard it was a really friendly community and like a good place to get your feet wet, so I went and did that, and I had a great time. I was an undergrad, so I kind of kept it, and I took some gap years, so I kind of kept it on the back burner for a little bit. But I knew that when I got into grad school, and I ended up coming to UCSD, and I came to Southern California, like one of the first things that I wanted to find was a community of environmental toxicologists, and try to like get back into some of that. And so actually through Google searches with this in mind is how I found SoCal SETAC. I also have to give credit where credit is due, because when I joined the program, I had an upperclassman, or like an older grad student mentor in Tara Wiggin, and she was quite active with SoCal SETAC for a while, and so she was really enthusiastic about like encouraging me to get involved with the community, and she brought me along to meetings and help get my feet wet there as well. And so I think like her enthusiasm for the community was kind of like contagious to me a little bit. And so it really was kind of from day one from grad school, from these two reasons that I got involved.
Let's see. Okay, last question. Do you have any advice for lay people looking to be better stewards of the environment based on your work or other people's work, different things that you've been thinking about lately?
Educate yourself. Don't be afraid of asking questions and trying to get answers on some of these important topics about your health and about the products that you're eating, and just so that you have all the information you need to make the right risk assessment for yourself about like what makes sense for you and your means and your lifestyle, but also like your health and concerns in your community that you're in. So